Top Bed Talk.
Hello, all right.
Good evening.
Look at you beautiful people. All right. Well, hello and welcome. My name is Rob Schiller, and I am a CA2 resident at Duke in Anaesthesia.
Bridger Bach, also a CA2 resident in Rob's class. We wanted to thank everyone for coming tonight. It's great to see such a great turnout of so many peri-optimists. And I love that that's a hashtag on Twitter. We also wanted to thank Top Med Talk for coming here and hosting such a great event, and also the Morpheus Consortium. So some people here might know what TRIPOM is, some people might not. So what TRIPOM is it's an international organization. Now, currently, it's based out of the UK. We also have branches in Australia, New Zealand. There's a branch starting up in Asia. And recently, we started up here in the United States. Now, our division is just about 1 year old. But so far, we have a growing base of trainees here. But we're always looking for more people. So we have 17 members of TRIPOM, and we're mostly based out of the Triangle area with hopes to expand across the United States. Now, the goal of our organization is to promote trainee involvement in perioperative medicine, and that's any trainees who come in contact with a patient from the moment they decide they want surgery throughout the perioperative period until the moment that they've recovered and they're okay to be handed back over to their primary care practitioner. So we're looking for surgical trainees, medical trainees, physical therapists, nutritionists, nurses, anyone who wants to be involved in the perioperative care of the patient. What we do is we produce educational material. So we have some tutorials online. We're also going to start producing a series of podcasts. And eventually down the road, we'd like to become participants in research in the perioperative community. So, do you have anything else, Ben?
I do. Albeit we are small right now. That's going to change. I promise you we're going to be big. We are part of a much larger organization, TRIPOM UK, which was started in the UK, started by our patriarch, Dr. Whittle, who you'll meet momentarily. And we are going to be expanding across the U.S., as Bridger mentioned. And we're very, very excited to be here and have your support and to be part of this momentous occasion. So without further ado, we're going to introduce you to Dr. John Whittle, who in our minds is absolutely famous, and if he's not yours, he will be momentarily. He is an assistant professor of anesthesia at Duke. He's also an intensivist. He trained in London at Guy's, King's, and St. Thomas Hospital of Medicine and the Central School of Anaesthesia. He's a fellow of the Royal College of Anaesthetists and faculty of intensive care medicine. Dr. Whittle has numerous publications and recently received a coveted Dream Innovation Grant to explore the role of low exercise capacity and parasympathetic function in surgical outcome. He is also the co-founder of TRIPOM UK, which we are part of, and also TRIPOM USA. His research interests are in perioperative optimization, including prehabilitation. So please welcome Dr. John Whittle.
Thanks. So you can already see question number 2 up there. The cool people have started. Without further ado, I'm going to tell you about the format of this evening. We're going to have we have a fantastic guest speaker, Dr. Molina, who I'll tell you about in a second. He's going to give a short talk about frailty and mitochondrial dysfunction. We're then going to have a short break for more liquid refreshment, etc., and then a panel discussion around topics that maybe have arisen from what Dr. Molina has said. To remind you, Slido is live. You can ask questions. If you're afraid to ask questions in the audience, don't worry, you can go up as anonymous. You can like people's questions, and that will bring them up to the top of the list. So without further ado, Dr. Molina is a gerontologist who has a research focus in bioenergetic dysfunction and function. His work is to promote healthy aging across 2 primary domains, cognitive function and physical ability. His team were the first to demonstrate that multiple features of aging, including physical and cognitive decline, are strongly associated with the bioenergetic profiles of circulating cells. I'm sure we may hear some more of that now. He serves as the Vice Chief of Research in the Division of Geriatrics and Gerontology at UCSD. That's a relatively recent move. I'm told he's also a surfer, so that fits in with our cool location, and remains an Associate Professor of Gerontology at Wake Forest. So thank you so much, Dr. Molina, for coming to talk. And if I'd like to invite you up to the stage.
Thank you. And thank you for the invitation to come and speak today. What I thought I would do for this short talk is really to focus on concepts rather than just bombarding you with data. I will show a little bit of data from publications, but to get the details on those, I'm going to point you to the publications themselves. So I want to start with something I think we can all agree on, which is that energy is fundamental. So for me as a cellular physiologist, when I think about energy, I think about this molecule, ATP. So you can think of ATP as the currency of cellular energy. So since, as we all know, energy cannot be created or destroyed, in the cells, energy can be stored and utilized by the making and breaking of chemical bonds, such as, I guess the pointer doesn't work, such as the bond pointed out here to the phosphate group. So as ATP is broken down to ADP by hydrolysis, this energy is coupled to anabolism. And then as ADP gets a new phosphate group to form ATP, this is coupled to catabolism. So I'm sure we all remember that most of our ATP comes from this organelle, mitochondria, through processes of oxidative phosphorylation. And mitochondria have gotten quite a bit of interest recently, including in pop culture, so with memes, Star Wars references, and now you can go on Amazon and get lots of really cheesy mitochondrial t-shirts as well. So my favorite is this one down here. If you don't believe in science, you mitochondria be a dum-dum. All right, but this depiction of mitochondria, I want to tell you tonight, is actually incorrect. And this depiction that we see in our textbooks and in each one of these t-shirts is incorrect because actually mitochondria are not jelly beans. Rather, they exist as an interconnected web of organelles, forming this network that's present throughout the cellular cytoplasm. So this is an individual Ca7 cell with the mitochondria stained in red. So in addition to forming this complex network throughout the cell, mitochondria are also highly dynamic. Mitochondria move continuously, and they move at 0.5 microns per second at the subcellular level. This is incredibly fast. Moreover, mitochondria undergo continuous cycles of fusion and fission that really mediate the overall health of this network. So mitochondria have also been gaining a lot of attention in research. And I think one good way to depict this for you is to ask PubMed to plot the number of publications per year that have the word mitochondria in the title. And this is the profile you get when you do that. So I start here at 1890, and this is when bioblasts, or life germs, were first described. And these were later found out to be mitochondria. 40s and 50s was really the age when we started to understand the biochemistry of mitochondria, understanding the role in respiration, understanding ATP, and also a new field of study called bioenergetics. And then in the 60s and 70s, though these lines aren't really corresponding to the graph, this was the age of mitochondrial genetics, when we realized that mitochondria have their own DNA. We understood the endosymbiotic theory of their origin, understood the maternal inheritance of mitochondria, and the presence of mitochondrial Eve. In the early 2000s, something interesting happened. New respirometry instruments came into the market that really brought on the field of translational bioenergetics research, where we started to understand the role of mitochondria in human health and disease. And now, what we see as potentially the next wave is the development of clinical applications for mitochondrial bioenergetics, including a new era that people have been calling mitochondrial medicine. As a gerontologist, I study the role of mitochondria in the aging process, and indeed mitochondria have been implicated throughout all stages of aging, from development, the onset and progression of morbidity and eventual mortality. Mitochondria have been implicated in multiple aspects of human health, including cognitive, metabolic, cardiovascular, physical, and psychiatric health. And now, mitochondria are actually known to be one of the biological hallmarks of aging, including other things that have been established to be involved in the aging process. Now, when I became a gerontologist, one of the first things I learned is this: the fact that two older adults who share the same age and disease burden can have dramatically different responses to intervention, different recovery, and different disease progression. So while patient heterogeneity is a challenge in all areas of medicine, it's a challenge that's magnified when you're working with older adult populations, which are likely more heterogeneous. So improving precision healthcare for older adults is really an imperative of my work. And in the area of precision healthcare, really a lot of this is centered around understanding the factors that mediate patient heterogeneity. Over the past few decades, precision medicine has really been focused on understanding how individual factors contribute to patient heterogeneity. These include factors such as genetics, environment, nutrition, and lifestyle. But the important thing to consider is that each of these all contribute to an individual's unique physiology. So nowadays in precision healthcare, there's a greater focus on searching for integrative, cumulative, and this is important for aging, and measurable functional outcomes that can inform on patient heterogeneity. And in my laboratory, we look at mitochondrial bioenergetics as such a measure. So our approach is quite simple. We basically implement measures of mitochondrial function that were initially developed for basic research and applying them in clinical research. So over the past several years, we've been learning how to measure mitochondrial function in different patient samples, including primary cells, tissues, as well as isolated mitochondria. So one of the first questions we addressed is simply, what do we measure? I mean, indeed, we can measure ATP, but as I mentioned, since ATP is both utilized and stored, it's not really going to tell us directly what's going on with mitochondria. Another approach is to measure mitochondrial membrane potential. So this refers to the electrochemical gradient that exists across the mitochondrial inner membrane. And alternatively, the third approach, and the one we favor, is looking at mitochondrial or cellular respiration. And this refers to something that's going on specifically at complex 4 of the electron transport chain. So as an electron enters complex 4, it binds with molecular oxygen to form water. And it turns out that by measuring this consumption of oxygen that's mediated by complex 4, we can get a very precise readout of the current of electrons that's passing to the electron transport chain. So if you're electrically inclined, you can think of mitochondrial membrane potential as a mitochondrial voltmeter and oxygen consumption as a mitochondrial ammeter. And really, mitochondrial oxygen consumption has been the gold standard measurement of mitochondrial function since these methodologies were first described by Chance and Williams way back in 1955, and are really basically still using these same approaches. So my lab is focused on human bioenergetic profiling, and we have a number of areas of research that we're currently involved in. One of the areas of research that we focus on is patient safety, using human bioenergetic profiling to see if we can identify whether an individual can safely undergo and benefit from a procedure or intervention. We also have projects focused on improving outcomes. Can treatment or rehabilitation be personalized based on an individual's unique physiology and unique capacity? We also have projects focused on disease prevention, identifying antecedent biomarkers that can be recognized so that we can undergo prevention before the manifestation of clinical symptoms. And then projects focused on cost efficiency, looking at post-hospitalization syndrome, and rehospitalization. I think the approach we've really been focused on recently, and what John was mentioning earlier, is blood-based bioenergetic profiling. All right, thanks. Okay, and this is based on the... Wow. Thanks. Okay. And this is based on the simple hypothesis that measures of mitochondrial function performed in circulating cells can integrate the cumulative effects of the individual factors I mentioned earlier, environment, genetics, nutrition, and lifestyle, on systemic bioenergetic capacity. And as you can imagine, blood-based profiling has a number of advantages. It's minimally invasive. You can do it in large-scale cohort studies, suitable for a wide array of patients, including the older adults that we work with. And this can be performed in a centralized facility where samples can be separated, prepared, and shipped out for analysis at a central location. Here's an example of what the current one of the blood-based bioenergetic profiling approaches we've been developing. This is a profile from a healthy older adult. We're taking peripheral blood mononuclear cells and measuring oxygen consumption rate, reporting on different entry points, providing nutrients to fuel oxidative phosphorylation of mitochondria, including fatty acid oxidation, and entry points into the electron transport chain. So the reason that reciprometric profiling can report on systemic bioenergetic capacity is because these cells are going to be continuously exposed to circulating factors that we already know mediate mitochondrial function across multiple organs. So in recent years, we've done some proof of concept studies to show that blood cells are indeed able to recapitulate the bioenergetic capacity of tissues of interest. So for example, we were able to report that blood cells can mirror differences in skeletal muscle bioenergetic capacity, shown here, where monocyte maximal respiration is significantly positively correlated to the respiratory capacity of muscle fibers and also isolated skeletal muscle mitochondria. Blood cell bioenergetic capacity is related to cardiac muscle mitochondrial function, and then also brain mitochondrial function. Here we looked at mitochondria that were isolated from the frontal cortex. Now, looking at brain metabolism in another way, we also reported that blood cells could mirror differences in brain glucose metabolism measured by FDG PET. So looking at glucose uptake in specific brain regions, we looked at here, frontal cortex, amygdala, and hippocampus. We saw that each of these was significantly positively correlated with the bioenergetic health of circulating monocytes. So in my lab, as John mentioned, we have two primary areas of research, cognition and AD, and also a program focused on physical ability and frailty. And for tonight's talk, I thought I would focus on our work in physical ability. And I think with this audience, I think you all know that there's a growing body of evidence linking physical function to surgical outcomes. For example, the publications show that preoperative physical function predicts self-reported quality of life. Physical function in and of itself is a key post-surgical outcome. And poor physical function predicts adverse outcomes after surgery. This was work by Dr. Groban, one of my colleagues at Wake Forest. So in order to demonstrate that blood-based bioenergetic profiling was related to physical function, we did this small study in 17 community-dwelling, overweight, obese, older adults, and focused on the physical function measurement of gait speed. So the reason we focus on gait speed is because to date, it's still one of the best predictors of morbidity and mortality in older adults. So this is a study that was published a while back by Stephanie Studensky, where she pulled data from 9 cohort studies, looking at almost 35,000 participants and almost 18,000 death events. And what she's plotted here for men and for women are the remaining years of life plotted by age group, with each of these lines depicting a different gait speed. So for example, a male at the age of 65 with a gait speed of 1.6 meters per second has on average a median survival of about 32 years, whereas a male of the same age with a gait speed of, let's say, 0.8 has a median survival of about 15 years. So what they calculated was a 10-year survival hazard ratio for every 0.1 meter per second decline in gait speed as 0.88. So that means a 12% increase in your likelihood of dying in 10 years for a physical function measure that's barely perceptible. So when we looked at these older adults, first we wanted to show that what we expected, that gait speed was significantly positively correlated with skeletal muscle bioenergetics. So here we looked at skeletal muscle mitochondrial function and found that with higher respiratory control, these individuals had better gait speed. But most strikingly, when we looked at blood-based bioenergetic profiling, we saw that these associations were even stronger. So looking at PBMCs, spare respiratory capacity and maximal respiration were both significantly positively correlated with gait speed, even when controlling for the individual's age, gender, and BMI. looking at other measures of physical function. We found that spare respiratory capacity of PBMCs, which I'm showing in these slides, is positively correlated with the short physical performance battery, knee strength, grip strength, as well as muscle quality, and again, when controlling for age, gender, and BMI. So, in addition to the studies that I talked about today, we do have a number of ongoing projects that I'm not going to describe, but I thought I would just briefly summarize. We have a study where we're utilizing machine learning to identify multi-parameter bioenergetic signatures, and this is really to advance our diagnostic and prognostic applications. We're also beginning to identify the mediators of systemic bioenergetic capacity. So remember again that the premise underlying blood-based bioenergetic profiling is that these cells are exposed to these circulating factors that mediate the bioenergetic capacity throughout the body. You also have studies focused on behavioral interventions that promote mitochondrial function. In particular, we're interested in diet-based interventions such as caloric restriction and also exercise. And then finally, with some industry partners, we're also beginning to screen for compounds that can promote mitochondrial health. So with that, I just need to acknowledge the people in the lab who actually do the work. This is our current Mito team and our generous funding support from the NIA and also the American Heart Association. And then finally, well, it's my email address and our website in case you're interested in learning more about what we do. Thank you. That is www.molinalab.com. AJ Molina at ucsd.edu. And we have multiple positions available at the moment if anyone's interested.
Thanks very much, Dr. Molina. No one jump ship and go to UCSD. If you could stick with us here at Duke, that would be great, and we'll collaborate. So we are just housekeeping. We're going to have a 15 to 20-minute break now, so recharge your glasses, have a think about what Dr. Molina has just talked about, maybe upload some questions to Slido, #TRIPOM19, and we'll have a short panel discussion afterwards with questions maybe more focused now on the perioperative period. So thank you very much. See you in a few minutes.
Top Med Talk. Thanks for downloading Top Med Talk. Let me just mention to you, EBPOM Dingle. From October the 7th through till the 9th, the 22nd Current Controversies in Anaesthesia and Perioperative Medicine and SIAA Autumn Congress will be live from Dingle Island, where it will be streamed around the world right to your home. EBPOM Dingle has a certain flavour about it. It's one of the conferences you'll have frequently heard guests and contributors on Top Med Talk refer to in hallowed terms. This is an incredible opportunity for you to be able to go there and enjoy every aspect of EBPOM Dingle that makes it so special. We've got the socialising covered, we've got all of the latest and greatest information, and we've got all of the fun you expect from an EBPOM conference. Check out ebpom.org, and I'll see you there.